Okay, well, thank you again to everyone who just joined. My name is Alex Mitchell. I'm a communications manager with the Lighthouse Lab Services marketing team, and we're very happy to have you all on with us today. Today, we'll be discussing culture versus PCR testing and the evolving lab testing market. We have a great panel, and we're really excited for you to hear some of their insight. I'll just quickly run down names and titles since background on everyone would probably be a bit much for us. We have Dr. Peter Bryant Greenwood, the Chief Medical Officer with Phase 2 Labs, Dr. Matthew Gombrich, CEO of Renaissance Diagnostics, Dr. Lori Milner, a Medical Director with the Lighthouse Lab Services team, Dr. Lindsay Stratman, a Medical Director also with the Lighthouse team, John Harrell, President of Lighthouse Lab Services, and Cassie Penn, a Client Manager for the Lighthouse RCM Solutions Department. And we want to thank them all for giving us their time and expert insight today. And with that, I'm going to turn it over to John Harrell.
Hey, everybody. Thank you very much, Alex. Excited to go through this topic today. We felt that this was really timely and relevant, and based on the interest level, we had 600 plus people sign up for this, and over 200 of you guys live today. We know that this is a conversation that's happening in laboratories around the country, and not only in laboratories, but in multiple different aspects of our industry as we try to figure out this balance and this migration a little bit of from culture to PCR testing. I know there's sales teams trying to figure out how to discuss this with physicians. There's physicians asking about how to incorporate this into their laboratories. There's MolDX and billing groups that are asking what is the value of that test, and that's really what drove us to come up with this topic and just get a conversation going around it, because I think that there's some pretty good consensus around the fact that PCR testing is really valuable and it does some things really well, and we're going to go through the pros and cons of comparing culture to PCR, but there's also questions about how to quantify that value, right? So as we look at the reimbursement for culture versus PCR, there's a pretty big delta there. And we're seeing pushback from MolDX and payers that are saying, okay, it's a faster test maybe, but how much more valuable is it? What is it doing to change how healthcare is provided? What's the value of that? What are the clinical outcomes? And so we felt like there was really a need to kind of come together and have a good conversation, just an understanding of the science at a base level will be presented today first, and then also a conversation around its place and appropriateness. I'm going to go ahead and push out an offer. I do feel there's a need for laboratories to work together on this issue. So we have formed a clinical laboratory advocacy coalition just to get laboratories together that are dealing with this, so that they can work together to help provide some of the support that's being required by payers. So some of the payers are asking questions about clinical utility and validity and citing sources and publications. And there's a heavy lift for any one laboratory to put together the documentation and research that's needed in order to respond to that appropriately. So we felt like it might be beneficial to see who's out there that wants to kind of lock arms and try to work on this together. There's no charge, of course, to join that. I think that'll probably look like a call every other week once we have a list of who's interested, but our first conversations can be about what that cadence looks like and how we meet to work together. So if you're interested in that, go ahead and click on that button, let us know, and we will go ahead and reach out to you with more information about how that coalition is going to work going forward. But I'd like to go ahead and turn over the control to Dr. Lori Milner, who's going to walk us through a comparison of culture versus PCR, the science, and the workflow. So Dr. Milner, it's all yours.
Thank you so much. And I am so passionate about this topic as a lab director that directs both a lab that uses molecular testing or PCR testing for UTI and a lab that uses traditional culture. This is just a very timely topic for me. So thank you very much. So just to really put culture in context of the workflow and actually what it is, what it's doing, and the differences between PCR and culture, just going to the history a little bit about it. So culture has been around for a long time. It was really first described in the 1800s by Robert Koch, who first published an article on the etiology of tuberculosis in 1882. So when we talk about culture, it's been used scientifically for quite some time. He was able to culture the first pathogen, the causative agent of tuberculosis, where he was able to culture a pure organism and actually recreate the disease in a susceptible host. It was then later better described for the current use that we use for it today for UTI in the 50s, when it was described, to discuss UTI in kind of the context that we use it today. So we provide both quantitation, a semi-quantitation, and identification. It is semi-quantitative in nature. It's not truly quantitative. We usually provide it in greater than 10 to the third colony forming units per mil. And then we are also able to provide phenotypic susceptibility testing. So this looks like we isolate a pathogen, and then we're able to determine if a specific antibiotic will destroy that pathogen or not. So looking at the overall workflow for culture, once a specimen comes into the lab, it is then accessioned, and it's then inoculated on one of those agar plates in kind of a petri dish. The goal of this inoculation is to get pure single pathogens that we can then just select one colony forming unit from that plate. So once it's inoculated, it is then incubated, and this occurs in an incubator that has a CO2 tank attached to it to provide a stable CO2 environment. The plate is allowed to incubate for typically 24 hours. It can be longer than that depending on how fastidious the organism is, but typically 24 hours, where we can then provide a preliminary identification in some cases. And then it goes on, typically an FDA cleared instrument that provides an identification as well as that phenotypic susceptibility to antibiotics. This whole process, depending on the instrument, every lab's a little bit different, depending on the instrument, is around 36 hours, depending on the instrumentation. So this is a longer turnaround time for this assay. What are some of the limitations of detecting urinary tract infections using standard urine culture? So one of the big things that we see is that in the literature kind of reports this as E. coli centric, or the media that we use, the nutrient broth media that we use is really good at growing E. coli and some of those other common UTI pathogens. And so that's where we get that term kind of E. coli centric culture. But for some more fastidious organisms or anaerobic organisms, we find that the PPV and NPV is really low. So we are not good at growing these in culture. Those really are the Enterococci family, group B strep, and those other anaerobic bacterias. In some instances, UTIs are polymicrobial in nature. And so there are multiple pathogens that might cause that infection. Culture it's not easy to do that for culture because we have to create those pure isolates. So that can involve subculturing and subculturing. So that can lead, to a very extended turnaround time to get those pure pathogens. So we typically don't report more than 2 pathogens using urine culture. The literature has stated that UTI is not an appropriate test when there's a high clinical suspicion of atypical organisms. And that is definitely seen when there's a physical urinary tract abnormality and those anaerobic bacteria grow. We're just not good at growing those types of organisms in culture. Additionally, as I mentioned, there is a long turnaround time for this assay, and it does require highly trained microbiologists and is labor intensive. So looking at just now the benefits and limitations of culture is still regarded as the standard of care, and those instruments are FDA cleared. Culture because in nature, how we're growing those organisms, we're only going to report an organism that's viable, that's capable of replicating. And we do also identify that specific organism with a phenotypic sensitivity or resistance. So as we'll see with PCR, that's different, where we don't separate the resistance. With culture, we are able to identify the specific pathogen that is expressing that phenotypic sensitivity. The cost of goods per test is inexpensive for culture, especially when we talk about the negatives. It's really cheap. So now some of the limitations. The turnaround time for culture is long. It does require that specialized setup and equipment. We've got the incubators that hold the CO2. You've got, microscopes for staining. Then you've got typically the instrument for sensitivity that can run, in most cases, sensitivity and that phenotypic susceptibility. It does require a highly trained staff, someone who knows what they're doing, knows what they're looking at. And then we have that limitation that we only report up to 2 organisms. So now let's look at a molecular or a PCR workflow. So again, once we get the sample into the lab, we receive it and we will accession it into the LIS. The first step is, well, in most cases, some labs don't extract, but most labs are still extracting from, if we're talking about UTI, then it's going to be from, urine. You're going to use that whole urine sample and extract typically a mil of it. That process can take up to about, depending on how many samples, but we'll say about an hour. Then for the real-time PCR plating and run, we'll allot, up to 3 hours for that or so, again, depending on the sample volume. And then we've got about 5 hours time to final report. So this is going to vary a little bit from lab to lab, but again, definitely possible to get that result out in the same day of receipt for sure. So now looking at the benefits and limitations of PCR. So when we're talking about PCR, we're detecting the presence of typically DNA, and it's not limited in the ability of the pathogen to grow on that media. So we're not growing anything, we are extracting what's there and identifying the pathogens that are present in that whole sample. Another benefit, again, is that the PCR can definitely be performed and results released in that same day. And PCR can result multiple pathogens without a problem. Some limitations of PCR are that you have to know what you're looking for. So the primers and probes must be included in that panel to detect pathogens or antibiotic resistance genes. Antibiotic resistance genes may not be equal to that phenotypic susceptibility that we do when we're looking at culture and sensitivity. So that can be, there are definitely times where it has been reported in the literature that those things aren't equal in every case. And then the last limitation that I'll mention here is that PCR detects the presence of DNA, and it doesn't necessarily differentiate between competent or live pathogen. So in some cases, if you're doing a straight qualitative assay, there may be no indication that there's very small amounts and that we have the potential to look at live or dead organisms. So then just comparing the two head to head. So as I mentioned, culture is still the standard of care, and there are those FDA cleared assays that can report this. Although for PCR, there is lots of emerging data demonstrating the usefulness, especially in certain cases. So that we're seeing in some cases in the literature, it's been reported that there are decreased hospitalizations when we look at culture versus PCR and diagnosing patients via the two methods. Culture is great, as I mentioned, at detecting E. coli and some other easy to grow organisms. But again, if you've got multimicrobial or fastidious organisms, culture really is limited to grow those out. Culture can identify resistance or susceptibility with a specific organism. So when you look at those traditional antibiograms, we're able to classify one pathogen with susceptibility. Whereas PCR, we're typically not separating out those resistances. So we'd report resistance for a sample in total. And for PCR, we're looking at antibiotic resistance genes, not phenotypic susceptibility. So those are different. Culture, as I mentioned, only reports viable organisms or organisms that are competent and able to replicate, where PCR detects the presence of DNA and doesn't discriminate between competent or incompetent pathogen. Culture is limited to those growth conditions on the plate, so it takes the right kind of organism to grow, because we're not using a human incubator, we're using a petri dish with media that's on that petri dish. Whereas PCR, as long as the primers and probes are there, we're not limited to the type of pathogen that we're able to detect. For culture, the turnaround time can be long, especially if you're having to culture and subculture. And then for PCR, those can typically be released on the same day. Culture is limited to those two pathogens, whereas PCR can easily report multimicrobial infections. And now I think I'm passing it back to the moderator.
Now we're going to hand it over to Cassie Penn with our RCM team to talk a bit about reimbursement comparisons.
Okay, as we look at the reimbursement, you can see that the standard culture to PCR is quite a bit lower. And we've seen this evolution. And I started out with stool and respiratory because those are PCR panels that have been around and are widely accepted by payers. And the difference with the GI and the respiratory panel compared to some of the other newer molecular panels out there is there are panel CPT codes. And there is policy out there. There's been LCDs out there on the medical necessity when it comes to those types of panels. The biggest problem when we look at these two panels is payers do not like to pay for more than 5 pathogens in a panel. And with stool cultures, I would say probably back to like 2015, a lot of labs did that migration where they stopped doing stool cultures and started doing the GI pathogen panels. So right now with these panels, really your fight is if your panel is greater than 5 pathogens, you're not going to get that reimbursement. Or with the new LCD, it really talks about for those expanded panels having a specialist order those. And with the stool and respiratory, there are FDA approved panels, sets out there, and it is highly used. Now, as we look at some of the other panels that have become pretty popular in the last couple years, urine, wound, nail, those panels do not have panel CPT codes. And those panels are really kind of just based on what the lab builds and what organisms are in there. And you can see as far as reimbursement, the PCR panels they can range anywhere probably from $100 to $600, maybe even more depending on how many pathogens you have in your panel. But your base cultures are, your typical $20 to $60. So really the ultimate thing that labs have to be able to do, and especially if they're in the MolDX Max, is they need to show the payers what is the value that PCR testing brings compared to culture. And as Dr. Milner had said, that culture is still considered standards of care. And so as you look at the new LCD, that's really what the MACs are trying to get through with their LCD is labs really need to have to try to show, especially if they're utilizing non-FDA panels, or if the panel doesn't have an FDA use, they really have to be able to show their analytical validity, and clinical utility, and really show the payers what that value is in that PCR testing. Spending more, what does that do for the patient? The other panels, the STI panels, your chlamydia, your gonorrhea, your trichomonas, those are pretty standard cultures and PCR panels. You're not seeing the chlamydia, gonorrhea cultures. You see the PCR panels. There are many payers that have policies out there that'll cover. They don't technically have a panel CPT code, but there are FDA platforms out there. There are FDA platforms out there for vaginitis. Those panels still do have some problem organisms in there that payers just don't like to pay for, but we are seeing some progress. And ultimately, really, as we look at it, it's being able to show that value of what that PCR is bringing for the patients. And now I will turn it back over to the moderator.
Thank you, Cassie. I think Dr. Stratman's going to talk a bit more about some of the validations and clinical utility studies that Cassie just referenced.
Thank you, Alex. So for these infectious disease panels that we are talking about, in order to be reimbursed for those, you need a Z code through MolDX if you are in one of their regions. So essentially, when you submit for the Z code, which is the first step, MolDX will determine if you need a technical assessment or not. When we are talking about UTI in this presentation, there are no FDA-approved tests out there that are around nucleic acid amplification tests. So it will be classified as an LDT, and you will be required to submit a technical assessment. Now, the technical assessment does consist of a lot of parts, but the main part, as I'm sure a lot of you all are aware of, is essentially that Excel sheet that is asking for a lot of information from your analytical validation. So that's kind of under that test information there. As you can see, so your specificity, sensitivity. I think one point of contention for a lot of us is that clinical accuracy, which I'll discuss a little bit more. But within that Excel sheet, it's also asking you for the literature, clinical validity, and utility. And so before I get into the next few slides, at this point, there have been no papers reference that MolDX has approved showing clinical validity and utility. But let's break down a little bit more what those three parts of the validations are. Now, the analytical validation is what, again, you all are really familiar with, what the majority of labs have done as required by CLIA, as well as other accrediting agencies, is how well does the test detect the mutation or compound that we're looking for? In this case, a lot of these infectious disease targets. So with that, again, I think that big point is that clinical accuracy and getting the correct number of clinical samples for each target within your panel. But where I think some of the confusion really starts to come in is that clinical validation and clinical utility. So here are just a couple definitions from the CLSI. These definitions kind of vary between agencies, but essentially with clinical validation, you're really seeing, is the accuracy of our test, is it predicting the presence or absence of that clinical condition? And so when MolDX is saying that, these publications don't meet clinical validity, they are usually putting them together with clinical utility. So we really want to separate that out. So in the point of the clinical validation, a lot of papers have been published showing, that comparing culture to the PCR, but not necessarily that clinical condition or outcome. So I know further studies are needed there. Again, to some of the points or limitations of PCR that Dr. Milner pointed out, that as far as determining cutoffs for, again, if the condition is there or not, especially when we're dealing with some of the PCR issues of not looking at viable or non-viable cells, we really need a little bit further studies there, or MolDX would like to see those. And as far as clinical utility, again, that value or benefit being assigned to a particular outcome, there's a lot of ways you can prove this, as shown in this picture that comes from CDC's website from the EGAP recommendations. But we can really look at this between, health risk, pilot, quality assurance, history, education, et cetera. But really what I've seen within the UTI is we're really trying to look at it from the economic perspective, that turnaround time, and that antibiotic stewardship. And there is a lack of two-armed randomized controlled trials there that we need to see. So that's part of, putting, again, that coalition that John mentioned at the beginning is really trying to get at some of those studies together a little bit further. So MolDX, it is important to note that they do require those clinical utility studies to be published literature. And that is kind of along the lines, I think, what they said in one of their talks once. Once you establish that chain of evidence that these tests, have that clinical utility, no one needs to reinvent the wheel. And again, just some other references that MolDX has pointed out. You have this EGAP, which was formerly the AACE. When it comes to, should you provide a clinical test or not, this is just a section of questions regarding clinical utility that they would like to see asked, and that really get, how do you design these randomized controlled 2-arm studies? So just kind of reiterating again some of the challenges I've mentioned throughout. When it comes to the clinical validity and really the analytical validity, each of those are going to need clinical comparator samples where we know a lot of that clinical information with those. And that can be really difficult for labs to obtain, especially some of these smaller labs. As far as clinical utility, again, a lot of papers have been rejected. We've not seen one that's met their acceptance criteria yet. As lab groups, a lot of our resources are limited to conducting these trials. Again, the need for coming together. And a lot of times labs have limited insight on those patient outcomes in order to get the information needed for those trials. So a few other challenges we've ran into is, again, just a few examples of accepted technical assessments. MolDX, has, again, that Excel document to fill out, but there is limited guidance there. And as, again, smaller labs not wanting to perform studies over and over, especially with that turnaround time, slightly more guidance, such as the FDA documents that are out there, would probably be helpful. I do realize they try to keep it general on purpose, because every lab does perform their analytical validations slightly different, according to each of those guidelines, whether it's a different contrived sample source that they're using, et cetera. It really is up to those individualized studies. So that's kind of what we're seeing in that MolDX space right now. And I'm sure there's several questions kind of surrounding this a little bit further, but I'll turn it back to the moderator for now.
Thank you, Dr. Schraubman. So I'm going to bring in Dr. Matthew Gombrich next. And so this is an example of kind of an alternative workflow. So hopefully you're starting to see the challenge that's in front of us as an industry, right? I think that we just haven't been thinking about it correctly. People have been worried about proving that the test works. Done a really good job with clinical validations, and we know PCR works, but what's being asked is, great, it works. It's even better, right? In some ways, it's faster. It can detect more. There's some benefits. Now help us quantify what that means in terms of clinical outcomes and what's the value of that, right? Because we're looking at rates right now where we see 10 to 100 times the reimbursement being requested when we're running these PCR tests versus culture tests. And have we demonstrated there's a 10 to 100 times value in, I think MolDX and most of the payers are saying no, that they don't see that yet. And so it's back on us to sort of look at how we charge for that, what the use is. Does everything get a PCR test? Should it be some other type of workflow in terms of how that is utilized? So Dr. Gombrich is going to come in and tell us a little bit about an alternative that he's been working on that's a sort of a hybrid or allows for some other workflow. So Matthew, if you don't mind, I'll go ahead and I'll turn it over to you to walk us through your solution.
Great, thank you. I appreciate it, and everybody at the Lighthouse team for inviting me. So I will tell everybody up front, I can't go into a lot of detail because of time requirements, obviously, but I'm happy to speak individually with people. You will see that what I'm proposing is a little bit of kind of the odd man out for this webinar, because my technology does not have to do with molecular applications, but kind of the upfront and the reflex that could happen with a positive test, both from our perspective, the platform, and a molecular into a more detailed rapid MIC AST test. So what my company, Reimbursement Diagnostics, has developed is an ultra rapid way of quantifying viable bacteria using digital flow cytometry. We have developed a tagging mechanism that enables us to distinguish the viable cells from the non-viable cells. And as Dr. Milner spoke about earlier, this is a big issue when it comes to using upfront PCR for diagnosing UTI in general. And our team thinks that one of the pushbacks from a clinical utility perspective and from a historical perspective, one of the reasons why there has not been more widespread adoption from a CPT reimbursement perspective is because the quantitative nature of UTI diagnosis is not addressed using molecular on its own. So we had been in some conversations with some laboratories that were using PCR for etiological assessment in urine specimens. And one of our statements back to these laboratories was, our data showed that upwards of 90% of the bacteria in a specimen submitted for suspicion of UTI was dead or injured and would not form a colony-forming unit. So we talked to these laboratories about the fact that our correlation coefficient on our system, and this is a sub 15-minute test that does both viable bacteria and white blood cells, our correlation coefficient was 0.99 to colony-forming units. And our workflow solution was one in which we thought you could screen with our methodology and reflex to PCR. And there seemed to have been a lot of interest. I've started a conversation with John and his team over at Lighthouse. I will tell you, we have done a pilot study with regards to diagnosing UTI directly out of the urine specimen, where we showed a sensitivity of 99.5% and a specificity of 91% compared to cultures. This is quantitative cultures. So our technology is ready to go. We have added a phenotypic antibiotic susceptibility profile. We don't call it an antibiotic susceptibility test for reasons that we can get into offline, but it's similar to the pooled phenotypic test that some laboratories are doing on Kirby Bauer plates. We have full intention of submitting our technology through the FDA. We have also applied, and this will get further down a couple slides, we have applied this exact same core technology to a detailed ultra-rapid MIC AST test, both that works in a McFarland standard and directly out of positive blood cultures, where we are getting data that is as accurate as traditional MIC panels in 2.5 hours with approximately 97% accuracy correlation. So the challenge for us is that the current guidelines and the clinical understanding isn't supporting the notion of a $300 to $500 reimbursement because of this quantitation issue. And so we're proposing a workflow that goes like this. Sample comes in, it's screened using our technology, the negatives are immediately reported back to the physician, and the positives reflex to the molecular PCR. A subculture would be done on the positivity of our test, and our detailed, what we call the mixed-stat rapid AST test would be done, all under existing CPT codes. So there is a CPT code for bacterial enumeration not using culture that reimburses at almost $30. And what we offer the end user is a 40 to 45% uptick on their cost for a $12 to $15 per sample uptick in reimbursement. The mixed-stat is a panel of 18 drugs currently. Right now, we have only assessed gram-negative pathogens, but we will soon be ordering our gram-positive panel. Again, a 35 to 45% uptick to the users. So what does that mean for the laboratories that would implement this? It's an upfront CapEx cost of approximately $350,000 for the instrument to perform this, but there is a $1.7 million reimbursement on the back end based on 450 samples a day. Our antibiotic susceptibility testing, capacity based on our modeling has about 113 positives per day with a $1.4 million CapEx cost, but a $2.2 million back end for the laboratories that implement this system. So we can go into more detail further on down the road. If somebody wants to reach out to me, I'm happy to talk in more detail about this and give a little technological background. But time requirements made it a little more difficult, but I'm happy to talk in more detail. Thank you.
Yes, thank you, Matthew. So an example of some creative solutions, right? So as we look at the cost for running a PCR for every test compared to a urine culture being a pretty big delta there, what are some alternative paths that can still provide some of that clinical benefit of the molecular testing, but without overburdening the healthcare system with cost is the goal. And I think it's going to take a lot of creative solutions, and I'm glad there's people working on it like Dr. Gombrich. So we will turn it over to get some questions here. I wanted to bring Dr. Peter Brian Greenwood into the conversation. If he had any comments, he sees this from a provider level. He does a lot of consults as a specialist in infectious disease. And I'm kind of curious, Dr. Brian Greenwood, would love to hear kind of your thoughts on this whole topic.
Yeah, thank you, John. I think that this is obviously a great presentation. I think there are some hidden gems right in this presentation that can provide a blueprint for any laboratory embarking on its clinical utility discussion, right, for reimbursement. And I just wanted to highlight some of those mentionable areas that I think are really worth emphasizing. Okay. Number one, several times, right, over and over again, culture is the gold standard. Culture is the gold standard, right? And so obviously in any kind of clinical validation or clinical utility discussion, providing data in absence of any kind of culture or correlation is going to be fraught with danger. Okay. So I think that's an important thing to realize, that the payers are being very savvy here and saying, I don't want to just see necessarily a molecular standalone solution to this problem. Okay. In other words, the first round of people who have the understanding that these two tests are rather complementary to each other is very helpful. Now, I think there's some ways around that if you don't have culture ready in your laboratory, okay? And that was a second thing that was brought up in this presentation, and that was who is ordering this test, right? And so anytime you're going to go down the clinical validation, clinical utility road, you have to ask yourselves, who's really ordering this test and what is it being used for? And the hint was given in the presentation, and that is the payers are very interested in obviously the specialists. And they didn't roll out of bed and say, well, we're just going to pin that on the specialists. They chose that because the clinical utility data exists not as a head-to-head with PCR and microbiology. You can PubMed until blood falls from your eyes, and you won't see anything out there with regard to head-to-head clinical utility, except in the context of specific disease states. So, for instance, I just did a quick Google, right? So you can find all kinds of clinical utility data around micro culture versus micro PCR in the context of COPD. So understanding, the disease states and leveraging your ICD-10 codes on those tests, on those requisitions, I think is really important, and communicating that to obviously the payers. So I'll be quiet and entertain questions, but John, thank you. I just wanted to emphasize that because that was very important, and I didn't want that to get lost.
Yes, thank you so much. And so I just threw that offer out there again for anybody that's interested in working together, but there's work to be done, as was highlighted. We're going to need people to publish for some of these disease states and really do the work and heavy lifting of figuring out how to compare those head to head and track those outcomes. It's a big lift. So, if you're interested in being part of that, please let us know. We will, obviously, anybody that participates will have access to that data to leverage for their laboratory. That way it's not on any one lab to do, but we're going to kind of reserve the rights to whoever's participating as part of the coalition will have access to the data, and we're not going to just share it publicly up front, or at least give first dibs to that, because I think there's work to be done and those people need to be rewarded for that. So I guess we'll get into some of the questions. Alex, I don't know if there was a few that you thought we should tackle first, or I can kind of look through the list and we can start throwing some of those questions out there.
Yeah, definitely. We could start with some of the ones that have gotten upvoted. Sort of a broad, future forward looking question, but Harry asks, what is the current future of PCR testing, specifically related to the current repurposing of the COVID PCR machines?
This is John, and I'll go ahead and I'll give my thoughts. This is just from the business case side of that, and then if there's any scientific or clinical thoughts to be added on, please pile on, guys. But I think that the payers were pretty savvy in recognizing that there was a lot of PCR instrumentation validated and out there in the market because of COVID, more than there ever has been. And as COVID dropped off, laboratories were going to look for ways to fill that instrumentation up with volume, right? And so as they started to look for how do we run these tests, what tests can this instrument run? Okay, it can run a urine culture, a wound panel, a respiratory panel, a gastro panel. Those sales forces were going to go try to find that sample volume, convert it potentially from microbiology over to PCR, and start filling up the equipment in those laboratories. And seeing, as you guys saw with those CPT codes that Cassie presented earlier, and that they might have paid $9 for urine culture and $900 for a urine PCR in some cases. That was probably a scary, and this is just me reading between the lines, but a scary idea to see this wave of tests that are going to come in at really high reimbursement numbers. And I think that's why we saw the MolDX LCD come out and the requirements for these C codes, and just to control what was happening. So I think that's the evolution. I do still think that PCR will have a place in the market. It's just going to need to get to a point where the payers are able to better measure the benefit, and they see that, yes, I would rather pay 2, 3, 5 times more because I'm going to get more. I'm going to maybe see my results a lot quicker, so the clinicians can act much quicker, and that's going to result in better clinical outcomes and cost savings in terms of the care to that patient. But we need to get there, and I don't think the data is quite there yet. Any other panelists have any thoughts or comments on the future of the PCR testing marketplace? I must have covered it all then.
Yeah,
why don't we go to
this is Pete again.
Sure.
I think on the future side, what we're looking at is understanding a couple things. One is what does the biofilm look like in this particular specimen? Clearly, when people are going down that clinical validation, clinical utility road, and you really want to front end a lot on the PCR side, I think there's a lot of interest in understanding not just, okay, I have a bug, I have a gene of resistance, but am I also looking at the presence of a organism that is producing biofilm? And that has certain very specific clinical treatment recommendations. So I think that's something that, again, only really PCR can provide that, because culture cannot, right? In fact, most of those non-planktonic forms don't even culture because they're just embedded in a mesh of biofilm. So that's one. And then the second thing is understanding when you're looking at things like wound, et cetera, are you looking at really a pro-inflammatory milieu, or are you looking at a pro-reparative milieu? And so many of the laboratories on this call may be looking to partner with, wound care, for instance, and they are obviously deploying some very novel and some very expensive wound care type modalities. And so I think that from a PCR standpoint, understanding biofilm and what kind of milieu you're looking at, I think is going to be important down the road.
That's great.
Yeah, thank you for that, Peter. John, I wanted to make sure real quick before we move on that we've got quite a few questions about MolDX Z code clarifications. Specifically, someone asking whether Z codes are necessary for tests apart from UTI. So I was hoping we could maybe touch on that again.
This is
Yeah, and I
Cass.
think Cassie, you're probably the best equipped to answer that one. Take it.
Yeah, I have the LCD pulled up, and basically what it says registered tests must, let me put it up here. Services that do not have Food and Drug Administration cleared and approved indicated uses, as well as FDA approved tests performed in ways not consistent with their intended use, will require a Z code. And that's what the LCD states. And so if it's not FDA approved or indicated uses, then it would require the Z code.
So all LDTs would require a Z code in the MolDX region.
Yes.
Is what I'm hearing there.
Yep.
All right. So hopefully that answers answers the question for most of you guys. It's not your favorite answer, I'm sure, but it is the one we've got. There was also a question, kind of how to talk to physicians, especially older school physicians, about the benefits of PCR testing. Many of them maybe have spent the last 20, 30 plus years ordering culture. How do you go about explaining the benefits of PCR testing to a physician that's traditionally ordered culture? Dr. Milner, you want to maybe take a run at that one? I know that, or Dr. Stratman, I know you guys both did a little bit of research, and I think you did present in your slides, or maybe just a quick circle back to that on the benefits of PCR and how you would maybe speak to a physician about it.
Yeah, this is Lori. So we do, when we have a consultation with a physician that's requesting information about the difference, if they see a PCR test that's new to them, then we would go through the explanation of kind of the difference between looking at the, in culture, you're seeing this whole pathogen that's growing on a plate, whereas PCR, we're just identifying what's in for UTI, for example, in like the raw urine sample. And then, we just go through some, the differences that they might see. So for PCR, they might expect to see more often those multimicrobial infections compared to culture that, typically only releases 2 pathogens. So we would just kind of describe the differences and how those methodologies are different, so that helps explain a little bit about why the reports are different. And then additionally, if they're looking at, depending on the type of report they see, the antibiotic resistances is typically the bigger kind of difference, because if they're used to seeing culture reports, they're used to seeing a functional sensitivity or phenotypic sensitivity, whereas a PCR report, depending on the type of report, again, may just release detection of those genes that may confer resistance. So we just kind of go over all the differences that they might see to help them interpret that data.
Yeah, and just to, sorry, this is Dr. Stratman, just to add to Dr. Milner's statements, we do see quite a difference in, final reports. Some groups are using places like ArcStone to help with that interpretation. And we do see that that's kind of helping on the side of, again, translating that information into, I would say, a more comprehensible final report. And that, again, is kind of more similar to those, antibiotics that we see traditionally with traditional culture.
Great. Yeah, thank you. Thank you both. Another thing that came up, and this was surprising to me when I had first heard of it too, was maybe it'd be a limitation of PCR testing in terms of its detection of possible non-viable pathogens or non-live pathogens, where culture is going to really represent those live viable pathogens. And maybe this would be a good one for you, Matthew, to tackle, because it sounds like in your research you saw that there was a pretty high percentage of that that was detected in PCR, but really wasn't viable or something that needed to be concerned with. Can you speak a little bit more about that?
Yeah. So the data that we've been deriving is from clinical samples. I mean, obviously we started on seeded, urine samples with, ATCC strains and things like this, but we've rapidly moved on to actual clinical samples. And what we have found is that in almost every sample, and maybe there's some bias or a caveat I should say after this, but in almost every sample, there is a substantial portion of dead or injured cells. So when we were using the automated flow cytometer, we knew that bacterial enumeration was the key for our success. And what we meant by that was not just, hey, there's a bacteria there, but hey, there's 10 bacteria there and 10 colonies showed up on the plate when we split the sample. As I alluded to in the few slides that I presented, our squared coefficient was 0.99 to colony forming units. Now, I've spoken to some experts in this area around molecular testing who have done some studies with clinical samples. I will say that our data may have been biased by two factors. One is the majority of our samples were from outpatient primary care offices, and some of the samples may have been older than 24 hours. We couldn't control for that. So does this data look better when we go side by side in a laboratory from the emergency department or the floors up above? Perhaps, but I can tell you emphatically that most urine samples have a lot of dead cells in them. So that's why we're advocating our workflow of quantify first, then in minutes, then reflex to the PCR, and then reflex to our mixed stat, which is the detailed MIC AST panel.
And I just wanted to jump in
Thank
here.
you so much.
Lindsay, I just wanted to say too, there's a lot of studies on what are like normal flora pathogens versus not. So I think that's just another thing to kind of add to the viable, non-viable, as, depending on the different patient populations, you might expect one pathogen to be present at a certain concentration. It might be when that pathogen decreases, or in some cases decreases on some women's health panels that provides, the clinical diagnosis information.
Yeah, that's helpful. Thank you. Another question that came in was regarding the Palmetto LCD and just asking, has anybody got a positive determination? I believe I've heard, and Cassie, I'm not sure if you have more information on this, but that there's been 3 approvals now, and that's been, I think, fairly recent that we've seen a few go through, and I believe it was LabCorp, Quest, and maybe one independent lab, but they were all for STI, if I recall correctly, and I think the rumor on the street is that it was around $150 reimbursement range. But Cassie, do you have any knowledge or thoughts on who's seen an approval or seen anything go through so far?
Yeah, it does look like there's been some, chlamydia, gonorrhea, some vaginitis, and from what I could see, there was LabCorp had some approvals, Quest had some, and there was one other one. I did not see, I kind of looked around to see if I could see more, but it looks like those panels are the ones that are starting to be approved. And you have to really work on your technical assessment and make sure that you're painting that picture of your validity, your clinical validity, and your clinical utility. I think the technical assessment is the meat to getting your codes approved. Even when you're looking at like the STI, that is one that's been around, chlamydia, gonorrhea, and there are some FDA approved panels out there. I just think you have to be able to show that picture in your technical assessment. And I think that's why there's only been, a handful of labs that have actually gotten their approval.
All right, thank you. Yeah, and I think most of the panels that we've seen groups apply for still have had no determinations outside of that STI area that at least from what I've seen. So thank you all for attending today. Our email address is on the screen. If you have questions, shoot that over. We'll do our best to answer those. Hope you will consider joining that coalition with your lab if you're interested in kind of locking arms and being part of part of the fight here. I think that there's a need for collaboration, and I think if we can move the ball forward here, rising tide, lifts all boats. And so I want to thank you for your time. I want to thank all of our speakers. Appreciate you giving some of your valuable time to help share some of the knowledge that's in your head. I know there was a lot of questions out there, and I think this was really helpful. So thank you all very much, and really appreciate your time today.